Hey, what's going on? It's me. And it is January 4th, 2022, and today we're going to go into the most well-known part of the immune system known as the antibody. Okay, so basically, what is an antibody? An antibody is a molecule, also known as an immunoglobulin, that is produced by our immune system that um, helps to tag and, in some cases, neutralize pathogens. Okay, so antibodies essentially they have a structure, all right, and like everybody knows about antibodies because you hear about things like this is an example of a COVID virus particle being tagged by antibodies. The COVID virus particle has characteristics on it that or antigens that identify it as COVID to your system, right? Because your system doesn't have the same senses. Your immune system doesn't have the same senses as people do, macroscopic senses. It's all chemically driven, right? Like at the microscopic level. So basically, an antibody is something that can bind to an antigen and thus allow a certain reaction to occur. Okay, so who what is like an antibody look like? Antibodies kind of look mostly all the same, right? There's different kinds of antibodies, so that's just one thing to note, right? And when I say kinds itself has a large grouping in it, right? Like there's different classes of antibodies, and then antibodies in each class can differentiate based on what they're attacking, right? Because there's like billions of different kinds of pathogens, right? So there has to be, since the antibodies help to identify pathogens, help the immune system identify pathogens, there has to be essentially billions of types of antibodies, but all the antibodies can be grouped into classes based on what they actually what kind of pathogen they're attacking, right? So, for example, you'll have antibodies that are essentially created. IgG, immunoglobulin G, is the antibody that is created so that it can help deal with your standard bacterial and viral pathogens. And you have different kinds of, like, you have IgM and other things like that, but then you have other kinds of antibodies. For example, IG. I believe it's IgE, which helps deal with large multicellular parasites, and it does that by allowing a helper T cell to create or to activate eosinophils. Okay, and eosinophils are cells which are specialized in dealing with parasites. But either way, like all these antibodies have structures which are kind of homologous across the spectrum of antibodies. So there's this Y shape here, and essentially an antibody structure. This is a better diagram for. I guess people to look at. So an antibody structure contains a heavy chain and light chains, right? Two heavy chains and two light chains, and the heavy chains are these internal chains here, right? Um, and the light chains in this diagram denoted by VL and CL, you can tell light chains are outside the heavy chains. The basic structure is that the heavy chains are bound to each other by disulfide bonds. So a disulfide bond is basically sort of like an intermediate strength uh, protein interchain bond, right? And if you've learned, your chemistry, your basic organic chemistry, you'll know that they are disulfide bonds have some level of strength, right? They're stronger than hydrogen bonds, not as strong as other covalent bonds or peptide bonds, but their strength level serves a specific purpose. And then the light chains are bound to the heavy chains also by a single disulfide bond, right? So again, but the regions. What you have to know is the regions of the antibodies are what are important. So at the very top of the antibody, on the left and right, on these on the tip of the heavy chain light chain, on externally facing Y branch, here is where you have your antigen binding site. Okay, so remember, your antigen is just some identifier that your pathogen has, right? Like, it could be anything. It could be like a long chain of lipopolysaccharides hanging off of a bacterial cell wall, or it could be just some, piece protein from a virus, a part of the extending off the protein sheath, right? Like, all these molecules, all these, you know, uh, agents, right, and pathogens and cells in general, they all have identifying markers for whatever reason, right? They've evolved these simple molecular markers to do different things, but the antigens, antibodies, through their antigen detection region, are essentially designed by your immune system to detect these markers and thus identify the pathogen. Okay, so yeah, so basically, this is how it binds to the antigen, and then basically the effector region of the antibody is where the antibody then connects to, for example, a cell or something like that, through a region on the cell, so that it can kind of like activate the cell, right? Like, so different cells in the immune system have like different activation states. Like, for the helper T cell, the helper T cells will get activated by macrophages that present the antigen that's bound to their MHC, major histocompatibility complex, to the helper T cell. And once the helper T cell is activated, then the helper T cell, in effect, will activate B cells, right? And then the B cells produce the antigens as well, right? So the B cells produce the antibodies, right? And carry the antibodies over to the pathogens. And in this case, the antibody that the B cells are carrying, I'm thinking in this case, is just referencing the IgG, immunoglobulin G, and then those carry the B cells attach rather, or surround pathogens and tag them, so macrophages can more easily identify them as invaders. So basically, there's a lot of tagging and like attaching to things that antibodies are essentially in the middle of, being the like. The primary actors in, right? They share the antibodies share some tagging overlap with the complement protein C3b, which acts as an opsonization tag on bacterial cells, for instance, or viral cells, and so that the complement system can also attack. Because the complement system, it's a weird twist of things. Antibodies tag, but they also activate they do activation of the complement system, and then the complement system also tags and tells macrophages to come by, but it also tells other complement molecules to come by, and there's a lot of like drilling of holes that they do, along with hole drilling is also something that's done. It's not only done by the membrane attack complex, but it's also done by the T cells, right? Killer T cells. So I mean, there's just a lot of overlap in function of the immune system. But essentially, that's kind of what antibodies are. Their whole point is to kind of like go around and do tagging and identification of antigens and let cells, immune cells, know essentially what to attack and to activate cells and give them information. So that's why antibodies are needed. So that's kind of a short video on that, and I hope that provided some useful info. I think I'll be able to, as I'm learning all this stuff, keep providing more and more useful info. All right, I'll see you guys later.